Thank you very much for inviting me. So it's a case study, and I have more questions than answer to propose. So I will explain the context of these phase 3 trials with two doses, low dose, high dose, and a placebo arm. So two comparisons, and also several endpoints. So a primary endpoint for the indication and a secondary endpoint to include into the label, to claim that into the label. And I will explain that to open the gate for testing the secondary endpoint, we need a separable multi-test procedure if we want really to be strict according to some recent publications. And then I will go through the different strategies we thought about and explain our proposal and ask questions. Next slide, please. So yeah, 3 treatment arms, low dose, high dose, and placebo. The primary endpoint is overall survival, and secondary endpoint is progression-free survival. So they are highly correlated, but you can also imagine something less correlated, like quality of life, patient-reported outcome as a secondary endpoint. It can also be an example. So we want to maintain the family-wise error rate of 2.5% one-sided, and we have a hierarchical logical restriction. Okay, so first we test overall survival for low dose and high dose, and if it is significant in the low dose, then we are allowed to test PFS for the low dose, and similarly for the high dose. Next slide. So we want to ensure that the family-wise error rate is equal to alpha or below. And we also want to maximize the statistical power. The problem is closed procedures like home, Hochberg, fallback, or Dunet cannot hierarchically open the gate to the testing of secondary endpoints. Or if you do that, it may a little bit inflate the alpha risk, the overall alpha risk. When you take everything into account, the family-wise error rate may be a little bit above alpha. And only separable multi-test procedures can be used as gatekeepers. So if you want, of course, I cannot explain that in 10 minutes, but you have two references from Dmitry Yemko and Tam Han, very nice to read. And the one from 2008, they demonstrate that in the context of parallel procedure. So you just need to reject one hypothesis in the first family, and then you go down to the second family, and they also demonstrate that in the serial gatekeeping. So you must reject all hypotheses, and then you can go down to the secondary endpoint. And in the latter publication in 2010, they also apply that in a logical restriction. And they proposed a solution, a mixture of procedures. I will explain that. Next slide. So that's I just wanted to explain what's a separable multi-test procedure. It's a procedure that does not exhaust alpha unless all hypotheses are true, and can be used as gatekeeper in place of like Bonferroni. So that's the separability condition. And so yeah, the most important point is Hochberg, home, fallback, and Dunet are not separable, but the author proposed to make them separable, building mixtures of procedures. So for that, you will take one procedure and another procedure. You will take a mixing function. And with this mixing function, you can make a procedure that is more powerful than Bonferroni, and that is separable. That satisfies the separability condition that helps to demonstrate that the family-wise error rate is always below alpha. So this is what is explained in these publications. Next slide. So we explained that to our colleagues. So originally, we have decided to adjust using Hochberg only. And when we found out about these publications, we explained that to our clinicians. And we wanted to use Bonferroni, so we avoid the problems. we have no more problems. Then they asked to translate that in terms of power, and they didn't want to lose the extra power afforded by Hochberg. It was an oncology trial, and the question was, what if we fall just in between, and we have something, a treatment effect that is significant using Hochberg, assuming the two effects for low dose and high dose are similar, and it is not rejected. The null hypothesis is not rejected using Bonferroni. So they didn't want to go through Bonferroni because our sample size was limited, and so we had a debate, we had a dilemma. Next slide. We have discussed 4 scenarios. So the first one is using Hochberg only. So we adjust the effect of the treatment on low dose versus placebo and high dose versus placebo using Hochberg. If it is significant for any of these arms, we go down to the secondary endpoint. So it maximizes the power as the pros, and the cons is that we can't be criticized based on this recent publication, as it does not properly open the gates for the secondary endpoint testing. So Bonferroni, you can't be criticized, but it does not maximize the power. Then we thought about using what is proposed in this paper, so using building a separable mixture of procedures. So a procedure based on a mixture of 2 procedures using a mixing function. It's better than Bonferroni. It opened the gate to the secondary endpoint. But the con is that it's not well recognized. I don't know how the agencies would have reacted to that. There is complexity, and also there is some freedom in choosing your mixing function. And the last option we chose, we were about to propose to submit to the agencies, is using Hochberg for the primary endpoint testing. Then if it is significant using Hochberg, you declare your trial is successful. And then you test your primary endpoint again using the Bonferroni procedure for the sake of opening the gate to the secondary endpoints. And so it may lead to an apparent contradiction where if you have something borderline significant for Hochberg and not for Bonferroni, you will declare my phase 3 trial is successful. However, I will not test the secondary endpoints. I can't, which is a very strange situation. Next slide. So yeah, this was our proposal. I just explained it. The original plan was to use Hochberg, and this has been accepted when we sought scientific advice. So my questions are, what does the EMA think, and also the audience, of these more complex testing strategies using mixture of approaches? What would the EMA recommend? Also, is it, so at the beginning, the first session, many questions were asked, and if I had the answer, I would be able to answer my own questions. So do we need to be really strict in controlling the family-wise error rate or not in this scenario? And is Hochberg sufficient? So, thank you.
Thank you. Then I kindly invite Franz to discuss this nice paper presentation.
So thanks, Patrick, for introducing me, and also many thanks for Vincent for sharing his case study with us. I guess it's quite complicated to present a case study from a company, and it was really interesting for me to see the thoughts, how this trial were set up. Next slide, please. So, I mean, we have learned a lot of new terms in Vincent's presentation, and I think it's quite confusing if you see all these terms. And in Austria, we sometimes say, we don't see the forest for the trees. I don't know if you have the same saying in English. So I think we have to step back for a moment and really look, okay, what are we really interested in? Next slide, please. So what we're really interested in the control of the patient risk. Peter told me this for many years, and now very slowly understand it, what is meant by this. So we shouldn't promise equations things which are not true. So in statistical terms, what we're interested in is the probability of at least one false positive claim should be smaller than 2.5%. So that's what we really want to have. Next slide, please. So what I really liked in Vincent's presentation is that he visualized the testing strategy. Because now all these testing strategies become extremely complicated, and it's difficult to see what the whole purpose is. And so he really illustrated it nicely that first the primary endpoint should be tested, and only if the primary endpoint of a specific dose is statistically significant, you can go on and test the secondary. So I really like this kind of presentation, and it really helps the investigators, also the members of ethics committees, maybe also regulators, to understand what's the main purpose. Next slide, please. So I started very slowly. So what if only primary endpoints were tested? So we want to compare the low dose against placebo, the high dose against placebo. We want to use the Hochberg procedures as we compare both dose groups against the common control. We know its test statistics will be positive correlated, so I guess we can agree on it. Everything should be fine. So that's okay. Let's go on. However, now we also want to test secondary endpoints, still strictly controlling the type 1 error rate. So let's start again very slowly. Let's assume for the moment all hypotheses are true. So we are comparing a placebo against a placebo, which means there's no difference in the primary endpoint, there's no difference in the secondary endpoint. Here we know everything is fine, and in statistical terms we'll call it weak type 1 error control is ensured. However, is it really a situation we have to care? Because what could be the case? Next slide, please. Some null hypotheses can be true, and some null hypotheses can be false, which means, luckily for the company, hopefully the alternative is true, which means we have a drug which does something. So for the moment, let's now assume that the true effect of the high dose is extremely large, which means we will always get a very small p-value. What does this mean? If you have a very, very small p-value, this means actually with the Hochberg procedure, we can always test the low dose at the full level alpha, which means already we have alpha % false positive claims. So we have already fully exhausted the level alpha. Now, Vincent introduced the next step that before we can test the secondary, we would test the primary endpoint at the Bonferroni corrected level, so over alpha half. Now, as the high dose has always a very small p-value, even this new boundary wouldn't change anything. Next slide, please. We would add alpha over half additional false positive claims, and here the problem comes in, which means, next slide, please. Here you can see we have a little problem, because next click, please. Because now the type 1 error is actually inflated with the proposed procedure. And now if we assume independence between the primary endpoint of the low dose and the secondary endpoint of the high dose, which is a reasonable assumption to start with, that two different endpoints in two different groups are independent, then we can show that the type 1 error, that multiple type 1 error rate is actually inflated up to 3.8%, which is slightly larger than 2.5%. So this procedure won't ensure a strict type 1 error control, or we can also, no strong type 1 error rate control is ensured. So that's the bad news with this procedure. Although it's quite complicated, still it will not control the multiple type 1 error rate. And that's exactly the thing what Catherine was mentioning in the first presentation. People are taking one reference, taking another publication, they're putting the things together, and then unfortunately, if you mix new things, it can happen like here that something new comes out, and then you have to check all the assumptions again. And that's, I think, is a very big problem we have in ethics committees, in regulatory agency, that it's very difficult to check such things within 1 hour, 2 hours time. Sometimes we don't have more time. So next slide, please. So what is my summary? So with your last question, whether it's a problem that we have a successful primary endpoint when no secondary endpoint is tested, I think this is no problem at all. If this wouldn't be a problem. I think the real problem comes in when we have a procedure which does not control the multiple type 1 error rate, and about this situation, the regulator should care more, at least for all important variables. And when discussing this case, I also read after some years the current points to consider guidelines on multiplicity again, and it really outlines all important principles nicely. And this guideline was for me sufficient to discuss this case study, and I concluded for myself, there's in principle no reason to open this guideline now, because all principles are already in it. And I would be very scared if the principles have changed within 10 years, because then something terrible would be wrong. So I'm really not sure if you should open the guideline. And I was really surprised to see that now also methods should be discussed in the guideline, because then a guideline will always be behind the current developments, because to open a guideline it takes 1-2 years, and to write the guideline will take 1-2 years. So by definition, it will always be 5 years behind the science. It's not possible to include all up-to-date methods. So my proposal would be to start a question and answer document, like it is done for the bioequivalence guideline, so you can quite easily add things when they come up. And what would be more worth doing is indication-specific guidance for which family of variables multiple type 1 error control is really needed. One click. Still, Inge, can you click? Still, I'm curious to see what we will see within the next hours, days, months, years. And also, I think it's good that the FDA will publish a guidance on multiplicity, because then we have another multiplicity problem. For me, it's good, because then I can pick just the things I like from this guideline and that guideline, which is a good thing, at least for me. Thanks.
Thank you very much. Vincent, any comment?
No, thank you very much. I'm just very happy about the work you did on my presentation. It's really helpful.
Goodie. Okay. Comments from the floor?
So thanks to you both. Vincent. I'm trying to understand the rationale for why you wanted to control the false positive error for an analysis of progression-free survival as a secondary endpoint when you have overall survival as the primary endpoint. There must have been a background discussion on the sort of the clinical relevance of the endpoints, the decision making, the pharmacology, and how the drug was going to have its effect on survival, for example. And I'd be interested to understand some of that, because if you see an effect on survival such that the study is a success, to not see an effect on progression would be remarkable, unusual. Yeah. So almost implicit on there being a survival effect is that you have a pharmacological effect on progression, probably of a magnitude that's no less. So is it necessary to go through the thinking around the control of the false positive error in this particular situation for this particular secondary endpoint?
Okay, it's a good question. Actually, I wanted to simplify the design, because this question of Hochberg and opening the gate for secondary endpoint is already very complex. But the testing strategy actually was overall survival, and then you go down and you split your alpha between PFS and biomarker, looking for an interaction with biomarkers. And then if PFS was successful, keeping 80% of the alpha for PFS and 20% of the alpha for biomarker. If PFS is successful, then we had to test PRO using the remaining alpha. So it was already a very complex. So for the sake of this presentation, I just wanted to ignore that. So, I mean, this was the idea. PFS is something our clinician thought it was good to add into the label. But behind that, there were also the biomarker strategy, and then also the patient-reported outcome that they were coming. again with another level of hierarchical. Yeah.
Yes, please.
So if I can make a couple of comments. One maybe the first one to address your question, Rob, as to why we do this, because it's a very similar situation I found myself in my company, which is AstraZeneca, incidentally, in oncology. And the reason we do that, because I agree, it's, if there's an effect on survival, well, if there wasn't an effect on PFS, well, you'd have to question your primary endpoint anyway, so why do we need a multiplicity adjustment? It comes back in reality. We do the same at AstraZeneca, this issue of false claims. If I want to make a claim on my secondary endpoints, PFS response rate, and certainly by FDA, we're being asked to come up with testing strategies for that. So that's the reality of why that's done. But what's underpinning that, and I think the thing that we need to debate today, seems to be this implicit assumption that we have to have strong control. That's one of the key. It's kind of, as I've got familiar with this area, that seems to be the assumption that all of this is predicated on, and specifically that it might be true that your primary endpoint was true. Because if that's not the case, and your primary endpoint, your null hypothesis is true, then there is no need, even under strong control, to adjust for any of your secondary endpoints. So what all of this is predicated on is that we think the concept of strong control is the right one, and I'm sure there's going to be differing views on that. But I think that's an important discussion we have to have during the day.
Yes, please.
As far as I'm concerned, I really think the strong control is the thing we need, because otherwise we would promise the patients too much, which is not true. We would promise there is a decrease in progression, although it isn't. So we have to look at the strong control.
But Franz, when you described the importance of strong control in that example, it was predicated on the independence between doses and between endpoints, if I understood. And that's not reality here. The survival and the PFS are going to be, they're not independent. I mean, that's clear. And even the dose levels are not going to be independent.
That's true, but the important point is why you still can rely on the independence, because you have the low dose group and the high dose group, and there the data will be independent anyway, and the correlation only comes in through the placebo group. So still, I think you're completely right. The actual type 1 will be slightly decreased, but not much. Still, there will be an enormous inflation.
Yes, Peter.
Well, I want to go back a little bit. I think when 1991, the paper on multiple testing in clinical trials was issued, we thought also about this issue. But honestly, I think the decision why the multiple control is so important was just a little heuristically, because you can't have any justification. It depends on the risk and everything. I think there's some sentence in like, it's important that you don't come out of the trial with at least one serious error, because the multiple test problem is a very complicated one. You have hypotheses and you have all constellation of decisions. There are so many constellations, so what you reduce the whole thing at least one. So you reduce the complexity of the decision problem to a very simplistic situation. And the problem, it works. It's one of the problems. It works. You can do things because in this high-dimensional space where 0 and 1 can be combined for any hypothesis, there are so many outcomes. So this is a parsimonious solution, has been a parsimonious solution, and it tells the people who look at the trial, well, we come out here, we give you a list of rejected hypotheses, and you can be quite certain that there is at least one wrong rejection only with the probability of alpha. I think it's a very heuristic explanation, because the problem is basically very complicated. The decision problem in the old dimensionality is highly complicated, and the use of such an error control is a vast reduction of the complexity to a simple measure.
Okay, thank you very much. Do we have any more comments? Martin?
Yeah, maybe also to this issue of the weak versus strong control. And I think even the term weak control is maybe somewhat misleading, because actually in most situations, besides the global null hypothesis, you have no control at all of the type 1 array if you use a procedure which has just weak control of type 1 array. You have then to really to look at the specific situation, and it may depend on the correlation and so on, what is the actual extent of the inflation of the type 1 rate. But I think really to settle just for a weak control would nearly give up a type 1 error control at all in a clinical trial if you just restrict you to one specific scenario where you want to control your error rates.
I think at these times we had this problem with the F-test breaking treatment. Experimental scientists tried to use a very strong treatment group into a multi-treatment trial. So they did always the F-test. So they had a certain effect with one clearly effective treatment in experimental medicine. And then they made the multiple comparisons without any adjustment, according to Fisher. And this was a story which was very, very frightening, because it was used to do. Experimentists used one treatment where they know this is the F-test breaker. This makes us the F-test. And then we can go on. I think this was our motivation, that weak control is not a good thing to use.